Fragile X syndrome presented by Sarah Holmes and Jillian Wins. Fragile X syndrome is a genetic condition caused by a mutation in the FMR1 gene on the X chromosome. Individuals with Fragile X are affected differently, many experiencing impacts on cognition, behavior, and development. It is common to see intellectual disabilities and autism spectrum disorder with Fragile X as well. To be discussed in depth later on, females do experience milder symptoms compared to males with Fragile X. There are other names that Fragile X is referred to, such as Fraxa syndrome, FXS, Marker X syndrome, and Martin-Bell syndrome. There are also other variations of Fragile X, such as Fragile X-associated primary ovarian insufficiency and Fragile X tremor association ataxia syndrome. Fragile X syndrome occurs as the result of a frameshift mutation on the FMR1 gene. In a normal sequence, a trinucleotide repeat called the CGG repeat is repeated approximately 5 to 40 times. In individuals with Fragile X, there are more than 200 CGG repeats. In the diagram, the difference between a normal sequence and a mutated sequence that causes Fragile X are shown. In a normal sequence, the CGG repeat is repeated a small number of times, allowing for the normal production of the protein FMRP after transcription and translation. Those with Fragile X contain many more of these trinucleotide repeats, represented by the yellow portion, resulting in the abnormal production of FMRP. Frameshift mutations occur when there are additional nucleotides added to the mRNA sequence during transcription. When the CGG repeat is repeated many times beyond what is normal, this causes changes in the gene and its function. The FMR1 gene is located on the X chromosome. As shown in the image, the FMR1 gene is one of many found in the chromosome. When this gene is not coded properly, changes to the chromosome occur. The FMR1 gene codes for a protein called the FMRP protein. The FMRP protein is important for synthesis of other proteins, neuroplasticity, cell-to-cell communication, and the transport of mRNA. In cells, the cell-to-cell connection is called the synapse, in which FMRP protein helps to develop. Cell-to-cell communication is lost when the FMRP protein is not produced, impacting brain function and development in the nervous system. The diagram to the right shows the difference in function when FMRP is produced versus when it is not. When the production of FMRP, cells are able to aid in spine stabilization, protein translation, spine growth, and actin polymerization. In the absence of FMRP, individuals with Fragile X still experience spine growth and translation of other proteins, but there is no ability for spine stabilization. Fragile X affects the FMR1 gene that is found in the X chromosome. The fragile site is where the FMR1 gene is located on the chromosome. Because of the sex chromosome difference in males and females, each population is affected differently. In both males and females, each of the chromosomes is normal except for the sex chromosomes, in which females have 2 X chromosomes and males only have 1. In females, because they have 2 X chromosomes, a mutation in only one of the 2 FMR1 genes is sufficient enough to cause Fragile X. In comparison, since males only have 1 X chromosome, a mutation in the only copy of the gene is sufficient. It is because of this that males experience more severe effects than females. Fragile X is an X-linked dominant disorder. X-linked dominant means that the mutation that causes the disorder is on the X chromosome, and if one chromosome is affected, the offspring will also be impacted. In the Punnett square, an unaffected mother is crossed with an affected father. Because it is X-linked dominant, the affected father will pass the mutation onto each of his daughters, and the sons will remain unaffected. There is a 100% chance that the offspring is a daughter that they will be affected, and a 0% chance that the father passes it to a son. In the Punnett square to the left, we see an affected mother and an unaffected father. This combination results in the mom having a 50% chance of passing Fragile X syndrome down to a male or female child. And the Punnett square to the right shows a cross between an affected mom and affected dad. This results in the dad passing it down to all potential daughters and a 75% possibility of a child being affected no matter the gender. This match also shows a 25% chance of a daughter being born with 2 affected X chromosomes. This pedigree shows the offspring of an affected female and an unaffected male as the first generation. The first generation contains an affected male and unaffected female, and in this case, 2 of the 4 offspring become carriers or affected by the disorder. The affected mother has a 50% chance of passing it on to one of her daughters and a 50% chance of passing it on to her son. Because the female is affected and heterozygous for this trait, her children have a 50-50 chance of inheriting the affected X chromosome. In the second generation, an unaffected female mates with an affected male, making a 100% chance that the disorder will be passed on to each of the daughters, but will not be passed on to either son.
Risk factors of having Fragile X includes a person's family history, and to be born with only one X chromosome can impact a person's likelihood of getting this disorder. In males, a mutation in one copy of the FMR gene is sufficient to cause Fragile X, whereas in females, a mutation in one of the 2 copies is sufficient to cause Fragile X, but because they have another copy of the FMR1 gene to use, the symptoms are not as severe. Females with Fragile X are also at risk for Fragile X-associated primary ovarian insufficiency, which affects ovaries' function from a range to mild to severe, with mild being premature menopause and severe symptoms resulting in premature ovarian failure and infertility. The signs and symptoms of Fragile X are portrayed through social behavior, developmental delays, and physical characteristics. Some social behavior that can hint at this disorder is the delay of development in nonverbal communication, arm flapping, and anxiety around new people. Males may have trouble with hyperactivity and aggression, while females tend to be shyer around new people and struggle with paying attention. Developmental delays are seen as nonverbal communication delays and not reaching early childhood development milestones like normal. Symptoms can range from mild to severe and is seen as attention deficit disorder, difficulty thinking, reasoning, or learning. Some physical characteristics would be abnormal facial features, broad forehead, and a prominent lower jaw. These physical features can become more prominent with age. The statistical data from the CDC about Fragile X syndrome shows it is the most common form of inherited intellectual disability, with 1 in every 4,000 men and 1 in every 8,000 women being affected. A study with 557 people with Fragile X syndrome from the ages of 3 to 21 showed that 4 out of 10 had Fragile X and autism spectrum disorder. And the picture to the right shows ethnicity and the percentage of likelihood that they have the premutation for Fragile X syndrome. The tests and diagnostics doctors may use to diagnose Fragile X are commonly called Fragile X CGG repeat analysis or Fragile X DNA tests. The first diagnostic genetic test for Fragile X happened in 1970 by observing the X sex chromosome of a male under a microscope. They found that it appeared to be fragile due to the ends of the chromosome being broken. Today, tests are more accurate and use biotechnologies like chromosomal microarray analysis, which examines genes and finds missing or extra pieces of genetic material. Exome sequencing, which reads through a portion of someone's gene sequence, or PCR, and PCR is also utilized in testing to reproduce more of the gene when receiving the initial sample. The image to the right shows carrier testing performed to see whether someone is a carrier for Fragile X. Current research Long-term complications of Fragile X can vary from person to person, but it is more common for males to have more severe complications. Some are more likely to suffer from medical problems like seizures, anxiety, depression, dementia, and sleep apnea. Unfortunately, at this time, there is no one actual treatment for Fragile X, but there are options for minimizing the symptoms. This could look like starting with approved remedies at a young age, having a supportive education, going to therapy, and regularly visiting a doctor or physician. Pharmaceuticals can also help with Fragile X symptoms like ADHD, aggression, obsessive compulsive disorder, and sleep issues. Current research on Fragile X looks into treatments and potential cures. The clinical trials being done by Fraxa Research Foundation are beneficial for finding what drugs work best. Because symptoms of Fragile X range so far, it's hard for scientists to find a cure that works for everyone. And these are the references that we use. The references include Medline Plus, Rare Diseases Info, NIH.gov, FAXA.org, NCHDNIH.gov, FragileX.org, and MyClevelandandClinic.org.